Thank you for joining our Debbie's Dream Foundation Curing Stomach Cancer webinar on the role of clinical trials for cancer patients. Clinical trials are the primary means by which new cancer treatments are developed, tested, and approved. In this webinar, you will learn why a clinical trial may be right for you, understand the different phases of and common terms used in clinical trials, and hear tips on talking to your doctor about your clinical trial options. I am Margaret, and I will be moderating today's webinar. I'm the Patient Resource Manager for Debbie's Dream Foundation Curing Stomach Cancer. I am a licensed marriage and family therapist and recently completed my doctorate. My clinical and research background is focused on working collaboratively with doctors, patients, their families, and members of a healthcare team. We would like to thank Genentech for providing funding to make this webinar possible. We would also like to thank our promotional partners, the American Association for Cancer Research, the Anti-Cancer Club, the Canadian Cancer Survivors Network, Cure Magazine, the Esophageal Cancer Action Network, Meals to Heal, and Patient Resource. You will be able to ask questions during this presentation. You can type your question into the white text box that appears on your screen. At the conclusion of the presentation, we will address questions as time allows. If we do not have time to address your question today, we will try to answer it during the next webinar taking place on November 4th at 12 noon Eastern Time. In addition, the recording of this webinar will be accessible on our website in approximately 1 week. First, I will introduce Debbie Zellman, who will share with you her journey and information about Debbie's Dream Foundation. Then Dr. Daniel Catenacci will present Demystifying Clinical Trials, and then there will be time for a question and answer session at the end. I would now like to introduce the president and founder of Debbie's Dream Foundation, Debbie Zellman.
Thank you so much, Mary Margaret, and welcome to everyone on the call. So I was diagnosed with stage 4 stomach cancer in April of 2008. I was 40 years old and the mother of 3 young children. I was married to a physician, and I was a practicing attorney with my own firm. When I was diagnosed, I had no risk factors for stomach cancer, and my symptoms were very vague. I was told that my chances of being alive in 5 years was only 4%. However, I can tell you that it has now been over 7 years. I have had 5 recurrences, and I am still a patient. Next slide. A year after I was diagnosed, I founded Debbie's Dream Foundation Curing Stomach Cancer, and we are now a member of the Deadliest Cancers Coalition, the Patient Equal Access Coalition, the State Patient Equal Access Coalition, and One Voice Against Cancer. I serve as a patient advocate on the NCI Esophagogastric Task Force, on the NCI Patient Advocacy Steering Committee, on the National Comprehensive Cancer Network Gastric and Esophageal Guidelines Committee. I serve on the Integration Panel for the CBMRP Department of Defense Peer Review Cancer Research Program. I am on the ASCO HER2 Testing Guidelines Project in gastric cancer, and I am a stakeholder for the American Cancer Society Research Program. However, I'm told that my most notable achievement is that I have appeared on Dr. Oz in a program about stomach cancer. Next slide. So here's some facts about stomach cancer. Stomach cancer is one of the leading causes of cancer death worldwide, second in men and fourth in women. Each year, nearly 1 million people worldwide will be diagnosed with stomach cancer, and approximately 700,000 will die. More than 24,000 Americans will be diagnosed with stomach cancer each year, and more than 11,000 will die. Stomach cancer is on the rise in young people, ages 25 to 39. However, per cancer death, stomach cancer receives the least amount of federal research funds of any cancer. When I was diagnosed, there were few resources and support services for stomach cancer patients, and most people knew very little about this deadly disease. Next slide. And that is exactly why Debbie's Dream Foundation is a 501c3 nonprofit dedicated to raising awareness about stomach cancer, advancing funding for research, and providing education and support internationally to patients, families, and caregivers. We seek as our ultimate goal to make the cure for stomach cancer a reality. Our website is debbiesdream.org. Next slide. We have achieved many milestones in just over 6 years of existence, including a free patient resource education program, abbreviated PrEP, which helps hundreds of patients, families, and caregivers worldwide by matching them with other survivors and caregivers using specific disease criteria like stage, biomarker, age, gender, role, and region. We also hold free symposia and webinars like this one. We have 28 chapters in the US, Canada, and Germany, with more forming every week. We have events across the US and Canada, and we have authorized $350,000 in research grant funding. We've also held 3 stomach cancer Capitol Hill advocacy days and the first ever Capitol Hill briefing on stomach cancer. As a direct result of our advocacy, stomach cancer is now included in a $50 million pot of money, along with 10 other cancers, provided by the Department of Defense Peer Reviewed Cancer Research Program. In addition, our website contains in-depth stomach cancer information, a lecture library, clinical trial information and matching service, a stomach cancer support community, blog topics and resources, all of which are completely translatable into more than 60 languages. And again, check out the website at debustream.org. Next slide. So here's a snapshot of the homepage of the website, and you can see it's a very easily navigable site with drop-down menus, and a lot of the information and support is right under that tab, as well as information about us, or events, or news, or how to take action, as well as shopping and donating. Next slide. So now I'm going to turn the webinar back over to our wonderful moderator, Mary Margaret Kilmeyer. Thank you.
Thank you so much, Debbie. As you can see on your screen, there's a brief summary of some of the events that are taking place across the country as well as internationally. Coming up on November 7th in Chicago, Illinois is our Chicago Stomach Cancer Education Symposium. This is also a live webcast that is free of charge for patients, caregivers, family members, as well as healthcare professionals to come and hear from leading experts in the field of gastric cancer to talk about issues that are relevant for patients. This, again, is a free event that is live webcast international, so if you're not able to attend or be in Chicago during the event, we encourage you to go to our events page and register for that webcast or register for the live version. The following day, on November 8th in New York, we will be holding a Night of Laughter. And the entire month of November is Curing Stomach Cancer Month. This is an excellent way to raise awareness about stomach cancer. If you would like to apply for your state to list Curing Stomach Cancer Month to help raise awareness and spread the word about stomach cancer, you can learn more about that under Take Action on our website. Also in November, on the 13th, is an International Stomach Cancer Teens Day. This is another great way to raise awareness or do a fundraising project through your company, local schools, or other organizations that would like to participate. And then in January, we will have our Miami Marathon and Half Marathon team participating again on January 24th, 2016. And here you will see some important contact information, including our new office address in Plantation, Florida. Debbie Stream Foundation is open Monday through Friday from 8 a.m. to 5 p.m. You'll also see here some important contact numbers as well as email addresses to reach our office. We will now start the presentation on demystifying clinical trials. Our presenter is Daniel Catenacci of University of Chicago. He is also a member of the Debbie Stream Foundation Medical Advisory Board. Dr. Catenacci is a medical oncologist and assistant professor at the University of Chicago, specializing in gastroesophageal cancers as a part of a multidisciplinary team. Dr. Catenacci is very involved in clinical research with the goal of developing new inhibitory drugs for the treatment of gastroesophageal cancer and other digestive malignancies. We're very pleased to have Dr. Catenacci with us today, as well as a part of the Debbie Stream Foundation. And I would like to turn the webinar over to Dr. Catenacci now.
Thank you very much. It's my pleasure to present the slide deck to you. So, demystifying clinical trials. Next slide, please. This is the outline that I will be going over. The first is just to do a little bit of background. Debbie did nicely, but I'll just point out a few things relevant to clinical research. And then the second point, we will focus on what is clinical research and what is a clinical trial. Some of the specifics about the terminology that you may come across, phase 1, phase 2, and phase 3. What are statistics and hypothesis testing, and what is random variation, and why are controls and placebo necessary in some cases. and then focus a little bit on some newer approaches that have been implemented in terms of fast-track approval for therapy and regulation. Then finally, we'll look at some of the other logistical things that you may come across in terms of clinical trials, including what does the line of therapy mean, what is trial eligibility and criteria that we go through, and then other things such as the cost, location, and the duration of treatments that you would receive on a clinical trial. And then we'll summarize what we talked about. Next slide, please. So this is a pictorial form of the stomach, and we're talking here about gastric cancer, as you can see there in the light blue, and also in the stage 4 setting, esophagogastric cancer, which are often included together in clinical trials. The reason for this is that biologically, they do behave pretty similarly, and in the stage 4 setting, they're treated similarly. So unfortunately, even if found early, both esophagogastric cancer and gastric cancer, after having surgery and even perioperative therapies with either chemotherapy or radiation therapy, there was a high recurrence rate. And so we're either talking about newly diagnosed stage 4 cancer, which is very common, or recurrent cancer after a prior surgery. Next slide. So as we heard earlier, some of the outcomes in terms of stage 4 cancer were we call median overall survival, which means the time point where 50% of patients are still with us and 50% are no longer with us. If we look at historical controls of just supportive care, which otherwise would not be recommended today, but looking back in time, the overall outcomes are approximately 3 to 6 months in terms of median overall survival. With our best chemotherapy treatments today in October of 2015, those outcomes can be improved to on the order of 10 to 12 months, median. And now you heard that patients can be on extreme ends of this median, and this is just a guideline to see that 50% mark, but there are longer-term survivors in the order of 2, 3, 4, and even 5+ years, as you just heard. So chemotherapy definitely improves the median overall survival time. But even more importantly, when chemotherapy is administered properly, it is palliative, which means that it improves quality of life, including, for example, decreasing pain and other symptoms from the cancer itself. So the demonstration of these improved outcomes came from clinical trials conducted over the last decades. These new treatments were compared to the standard approaches at the given time and then replaced those standards to become new standards. Another recent example is HER2-positive cancers, which you, of course, probably have heard of, and now that we treat with trastuzumab or Herceptin, and that was based on a large randomized phase 3 clinical trial showing that the addition of trastuzumab to chemotherapy was better than the previous standard of chemotherapy alone. Next slide, please. So this is how we currently approach advanced or stage 4 gastroesophageal cancer, and we test for HER2 right away. As you can see, the majority of patients are not HER2 positive, but if you are, you get the addition of an anti-HER2 agent, trastuzumab. Next slide, please. So clearly there have been benefits, but clearly more work needs to be done to improve that median survival time. And so to improve or to prove a new therapy, it must be with a clinical trial. Now, why is that? Next. Well, you'll see throughout this talk that we, as physicians, take an oath when we finish medical school that we will first do no harm. And all of the intentions of clinical trials are to do just that. First, do no harm while we are trying to improve and provide benefit. And that's a scroll from the original Hippocrates in 3rd or 4th century BC. Next slide, please. So what is a clinical trial and what is research? A clinical trial is an experiment designed to answer specific questions about biomedical or behavioral interventions, including new treatments like drugs for cancer. They're only conducted after it's been approved, the protocol has been approved by health authorities and ethics committees, which, for example, are the IRB, or which you've heard of, which means an institutional review board, which includes a panel of physicians and other lay people to ensure that the proposed protocol is within ethical standards. They act by vetting the risk-benefit ratio of the trial. So if it is determined that the benefit outweighs the risk, then the trial is feasible and reasonable to proceed. It, of course, requires an informed consent of the patient and a written signed consent. Even chemotherapy that is not part of a clinical trial, as an aside, we at our center and many other centers require informed consent anyways, just to know what it is that we're getting into with just standard chemotherapy. The informed consent for the trial is just going into the specifics about the investigational agent that is being studied. A sponsor of the trial is someone or a group or an entity that sponsors the trial. So this can be a governmental organization, so there are clinical trials sponsored by NCI or the National Cancer Institute. It can be the academic center or principal investigator that is treating you, or it can be a commercial company like a pharmaceutical company or other companies for their specific thing that they are testing. Next slide, please. So what is phase 1, 2, and 3? So let's go into this. So first, any novel agent, so in this setting of cancer, usually we're talking about drugs. So in preclinical setting, meaning before it even is used in humans, drugs are being tested in cancer cell lines and animal models, and those that are showing promise over the years are then decided and brought into human studies. So the first human studies, or first-in-human studies, are called phase 1 for that reason. They are traditionally usually small studies, less than 20 patients, and the primary reason for existing is to seek to determine safety and tolerability of this new drug, the investigational agent. The goal is to start low and to titrate doses upward to ensure safety, of course, and this is usually done by enrolling 3 patients at a time and then closing. So if you're waiting for phase 1 trials, sometimes they tell you that it's not available anymore. That's because they open and close pretty routinely, because once 3 patients are enrolled, it closes and patients are followed for at least a month to ensure safety before resuming the cohorts to enrollment and increasing the dose, for example. Again, the goal is to find a maximum dose that is tolerated and ultimately to help decide what is the dose that the drug will be used going forward and used in the next setting of trials, like in the phase 2 setting. So for the patient, of course, the reason why the patient and physician would proceed to go on this type of phase 1 trial is in hopes to deriving benefit. But if the trial itself's endpoint is not that, the benefit for patients is considered a secondary endpoint in a phase 1 trial. Therefore, phase 1 trials are not usually done until other standard options or trials in later stages of development are exhausted. So the last point is important, is that it may not necessarily be a first-in-human approach in a phase 1 setting, but it may be, for example, the first time a drug is combined with another drug, and hence we're going, quote, back to the drawing board with a phase 1 study to ensure safety now that we're doing 2 drugs together. Next slide, please. When a safe and tolerable dose is established, the new investigational drug will proceed to phase 2. So phase 2 trials can typically range between 60 to 200 patients in size. An example of a small pilot study of 60 patients without any randomized control, and just everybody gets the investigational treatment, as opposed to a larger randomized phase 2 study, for example, that has 100 patients in the investigational arm and 100 patients in the control arm. So regardless, once the target number of patients is met, the trial will close to accrual. It successfully accrues and the trial then closes. So the primary reason for a phase 2 trial, which we call primary endpoint, is efficacy or benefit. And this can be a range of different outcomes, but usually is overall survival or progression-free survival, which means the time it takes before the therapy stops working. And secondary endpoints of phase 2 trials are always to continue to assess and monitor for side effects and the tolerability of the drug, and also to potentially determine if there are just subsets within gastric cancer, gastroesophageal cancer, that might benefit, whereas the whole population may not. For example, thinking back to HER2 positivity. So phase 2 trial act as a checkpoint to determine if it is a good idea to proceed to a larger phase 3 trial. And the reason for that is that trials are expensive and they're time consuming, and it can take several years for just the phase 3 trial to enroll, to follow, and to give outcome. And so we would like to be able to determine as soon as possible if a drug is not effective, to avoid going to these larger trials that take a long time, and to expose as few patients as possible to a treatment if we can find out that it's ineffective, and they can be better served with other treatments. So next slide. If a phase 2 trial is positive, though, that means it met its predefined statistical endpoints of benefit, then the sponsor may decide to proceed to a phase 3. Phase 3 trials typically range from 200 to even more than 1,000 patients, and are usually randomized and have an appropriate control arm. And we're going to talk about a little bit about what a control arm is in the next few slides. Before phase 3 trials are embarked on, sponsors usually negotiate with regulatory agents, either the FDA or the EMA, which is the European version of FDA, prior to finalizing the protocol, so that everything is in place for registration purposes if the trial is positive. In other words, it would become a new standard of care if it was a positive trial. And that's the last point there. So next slide, please. So what are statistics and hypothesis testing, and why do we randomize? So randomization, it's never a good thing, especially from the patient's view, and that is totally understood by all of us. However, there are reasons for it, and the randomization to a standard control is required to control for a number of known and even unknown factors, which we call confounders, that may affect outcome, like survival or whatever the outcome is that we're looking for. So example of known confounders are age. If you have a younger age compared to older age, that alone is a difference in prognosis. And so, and you can see other things like gender, pathological histology, which you may have come across in your pathology reports with well-differentiated or poorly differentiated, HER2 status, etc. So in the setting of a one-arm trial where there is no control arm, and it just happened to enroll a number of patients with all good or majority of good prognostic factors, it would seem to be a very successful outcome compared to historical controls. While it really isn't better, but it just happened to have all these patients that had very good prognostic confounders. So randomization ensures that an equal number of patients with good and bad prognostic factors are in each treatment arm. And then, if there is still a benefit in the investigational drug, we can be more certain that that benefit was from the drug itself rather than a number of these embedded prognostic factors which we may or may not recognize. So what is random variation? Randomization also controls for random variation, and I went through an example of how to understand what random variation is. If you think about flipping a coin between heads and tails 100 times, we know that the truth should be that you see heads 50 times and tails 50 times. But sometimes if you actually sit and do that experiment, sometimes you'll have 49 heads and 51 tails. And sometimes you'll have 53 heads and 47 tails, even though we know the truth should be 50-50. And sometimes, even rarely, you might even see 35 that should be 65. So even though it's unlikely, it's possible. So that is what random variation is. So randomization in a clinical trial setting ensures that if we see an improvement in the investigational arm, that it's real and that it's not due to mere random variation. Another example, realistic example with, say for example, median overall survival. If we knew that median overall survival was 11 months, and in this particular clinical trial that was only one arm was 14 months. If we don't have a control, we don't know if that was due to random variation as well as the other things above that we talked about, which are those prognostic factors. And so without those controls, we can't be certain that that treatment benefit is real. The last point is an important one, where randomization can be 1-to-1 ratio, which means that as patients come in, ultimately half the patients will get investigational treatment and half will get control. However, there are other approaches, and that can include randomization in favor of the investigational arm to try and get as many patients on the investigational arm. So in this example, I used 3-to-1 randomization, meaning that for every 4 patients enrolled, 3 get the investigational treatment and 1 gets placebo. This increases the odds of getting the drug, obviously, and makes participation much more appealing to patients, obviously. There is a requirement, though, in this type of strategy that statistically requires more total patients enrolled. So there's always a balance between this uneven randomization approach and the total number of patients required. And we talked about how trials can be long and expensive, and so if you need a large number of patients, it takes a long time to get the answers. So there's a balance between doing these types of strategies, but you may come across a 2-to-1 randomization in favor of the investigational arm. Next slide, please. So what about the placebo? The placebo, of course, is one of the notorious things that we don't like to have to do. But why do we do it? So placebo, or blinded studies, is a control arm that is placebo-controlled. And control arms don't necessarily have to be placebo-controlled, but in the setting of when it is controlled with a placebo, there's reasons for it. Placebo can control for many biases that occur if we don't do this. Well-recognized biases include physician bias. So the physicians want the drug to succeed. And so if we know certain patients are getting the investigational drug, or we're unblinded to that information, and there are equivocal decisions, which I describe here as an example of CT scan results. So sometimes CT scans require some subjective input of the physician. And if the physician knows that this patient happens to be on the investigational drug, maybe subconsciously they will say, well, let's keep them on and consider it stable disease. As opposed to if the patient was on the control, they may say, well, let's call this progressive disease. And similarly, with patient bias, if a patient knows they are on the investigational drug as opposed to the control arm, then they may report better outcomes in questionnaires, etc., that are describing pain and quality of life, etc. This is a well-known effect called the placebo effect. So by blinding or double-blinded, which means both the physician and the patient do not know if they're on the investigational drug or the placebo, then these types of biases can be eliminated, and if there is a benefit realized in the trial, then we can be certain that it is a real benefit. And then the final point, which I starred and bolded, is that it's sometimes a misconception, is that placebo-controlled arms will always be getting a standard of care plus the placebo, and never be deprived of known effective treatments for a trial in a given setting. So that is why we have these ethical committees, etc., to ensure that these types of things are enforced. But placebo controls are just matching to the investigational drug in the other arm. So for example, in the setting of the HER2 trial that I mentioned earlier, which was a large phase 3 trial, patients got chemotherapy with a control arm placebo or with Herceptin. So they were always getting standard chemotherapy with either the control, the placebo, or the Herceptin drug. Next slide. So one slide here just to go through Fasttrack Approval. So we've talked about going from preclinical setting to phase 1 to phase 2 to phase 3 and then to becoming a standard therapy if it meets all the endpoints of all those different layers. And that can take a long time. And so sometimes a benefit of a drug is so large that in, say, for example, the phase 1 setting or early phase 2 setting, the benefits are so dramatic that the drug can leapfrog a number of these steps and become accepted as a standard therapy or fast track. So examples include in other cancers like lung cancer that have a specific gene problem called ALK translocation, where the response rates were so high and unusual for that cancer that it was approved pretty quickly by the FDA. Another example more recently is the immune therapy, anti-PD-L1 and anti-PD-1 therapies that are being approved in just even last week in lung cancer with very few patients because the benefits were deemed so much better than what would be expected. And so this has direct relevance with gastric and gastroesophageal junction cancer because there are a number of trials being done and that we've participated in already in various stages of the disease. Next slide. So some other final logistical considerations that you might come across are trial eligibility. What is that? So eligibility criteria must be met after the patient provides written consent or signed consent to participate in a clinical trial. So there's a number of different eligibility criteria, but some common things include laboratory values. So your white blood cell count has to be greater than a certain number. Your platelets have to be greater than a certain number. You have to have stomach cancer, or you have to have a certain type of stomach cancer. You have to be a certain stage. So you have to be stage 4, for example, or you must be locally advanced, not stage 4, but not stage 1, if you're going to go to surgery to be involved in this perioperative chemotherapy trial, as an example. What is line of therapy? So a line of therapy is basically if you're newly diagnosed and you're getting chemotherapy the first time, that would be considered first-line therapy. If that line of therapy stops working and you get a different chemotherapy regimen, then you'd be second-line therapy, etc. So a line of therapy is another eligibility criteria of clinical trials. For example, some trials are only for newly diagnosed cancers that have never been treated before. Other trials are only for patients who've had 2 lines of therapy, so you have to be third line, for example, etc. So something to keep in mind is that even just 1 dose of chemotherapy at the time or just after you were diagnosed, and then you go and you find out about a clinical trial that's in first line, that would exclude you from being eligible for the trial. And this is an important one to recognize, is that many times patients come in and they've had their 1 dose of chemo and they're entertaining the possibility of clinical trials, but we can't include in first-line trials because of that 1 dose of chemotherapy. So that's an important thing to consider. Next slide, please. What about duration of therapy? This is a common question. How long will I be on the trial? Well, usually it is, as in most of our cancer treatments, we keep on it until it stops working, or until the terminology would be until disease progression. So some trials, on the other hand, do stipulate a time frame, for example, up to 24 months, and then you'll be taken off trial. So these are things to look into in the consent form and discussion with your physician when you're considering a trial. I think the rationale of some of these trials saying up to 24 months is that that is substantially longer, as we've talked about, than what would be expected. And if a drug truly is beneficial, at that point, potentially might even be already approved by the FDA and then could be done as a standard therapy. What about the location of therapy? Where are we going to get treatment? So because there is a lot involved with the trial in terms of data collection, lab draws, research studies, the uniformity of the treatment is also very desirable to limit that variation that we talked about. Therefore, treatments are required to be given at the enrolling site. And so this is not locally for most of the time for many patients, unfortunately, which is obviously more convenient for them. However, something you could look into is that several of these trial sites and academic centers do sometimes have satellite practices that participate in enrollment. For example, we at the University of Chicago have a satellite site at Silver Cross and Joliet that allows us to enroll patients and get treated closer to home. Next slide. And finally here, so another one is, what about the cost? What will this cost me on therapy? So the answer usually is found in the consent form, and it usually is that all standard visits and treatments are billed in the usual way to third-party payer insurance, and you may be responsible for copays as you normally would be. These include MD visits, laboratories, CT scans, etc. All of that would be under the umbrella of routine standard care. Anything, on the other hand, that would be considered research, the drug itself, any research labs that might be done, any research scanning that might be done, those are typically included in the trial and are not billed to insurance, but rather billed to a clinical trial fund that the sponsor has provided to the investigators. Next slide. So then to summarize, clinical trials are used to determine how we can advance our understanding and how to best treat patients. It is the only way to scientifically determine with statistical scrutiny that the new therapy is in fact beneficial and worthwhile using as a standard therapy. And our philosophy, as I talked about earlier, first do no harm, why would we want to expose patients to a drug that is expensive and potentially have side effects if we can prove it doesn't work any better than current practice? The clinical trial is a safeguard to prevent new agents from becoming standard that actually don't work. And this is what differentiates Western medical practice from all the other various products you can find on Google and through the grapevine, because those approaches are not rigorously tested. Next slide. I'll conclude there, and I'd be happy to answer any questions.
Thank you so much, Dan, for that excellent presentation and overview of clinical trials. I will now read some of the questions that we received during your presentation. The first, what constitutes informed consent? Is it a document or a conversation?
It is actually both. We sit down with the patient, usually it's the principal investigator or a research nurse or coordinator, that there is a document that has all of the information about the specifics of the trial, much of the stuff that we talked about here in terms of duration of therapy, cost of therapy, etc. Talks about potential side effects that might be known or unknown at the time, and everything that would be involved in terms of the scheduling and the treatment frequency, etc. of the trial. And after going through the whole document together, there's an actual signature page at the back of the form, at the back of the packet, in which you sign, date, and both parties would sign a date and say, yes, we discussed this, and we are prepared to move forward with this particular clinical trial.
Okay, thank you. And another question, do all trials examine the various types of endpoints, or are they designed to answer a specific question?
So we talked about primary endpoints and secondary endpoints. Usually a clinical trial will have a primary endpoint. So depending on the phase of the trial, the phase 1 trial is typically looking at safety and the dosing regimen and trying to get an accurate dose that's safe. Phase 2 trials typically are their primary endpoint as early efficacy to see if it meets that first check whether or not we should continue developing this drug. And phase 3 trials are usually looking at their primary endpoint at overall survival or at minimum progression-free survival. That's not to say that there won't be a number of secondary endpoints in any of these different phases. Secondary endpoints of phase 1 are efficacy. Does it show a nice response rate? In the 20 patients enrolled, how many patients actually benefited from this? Is it worth going for the phase 2 trial if 0% of patients responded, even if it's safe, as an example? And similarly in phase 2. Response rates, progression-free survival, and even in the phase 2 and phase 3 settings, secondary endpoints are always chronicling all the side effects, etc., to really, at the end of all of this, let us weigh the risks and benefits, the advantages and the disadvantages, and see if this is worthwhile to approve, and if approved, is it worthwhile for the individual patient after talking with their physician who knows these data to say, is it right for you? So that's why all through these phases of trials, all of these things are being accumulated, but statistically, the primary endpoint might be a little bit different.
And a follow-up question, if the primary endpoint is different, or negative rather, but there's something else that looks good in the trial, but it wasn't necessarily the question, would the trial be considered positive or negative?
That's an excellent question, and we alluded that to one of the slides where we said sometimes we look at subsets that may have benefited from the drug even if the overall population appeared to not benefit. So that overall population in our terminology is called the intention to treat analysis. And so typically the trial itself would be called negative in the situation you described, but cohort X, HER2 positives, or well-differentiated cancers, or any subset of choice appear to derive some benefit, for example. Typically, what we would then recommend is to say, usually the FDA would not approve the drug for that subset because that wasn't the primary endpoint, and usually that's a much smaller number of patients, and so that is subject to more variation and random variation that we talked about. And so what the official recommendation would be in that scenario is, now if you really feel that that subset was benefiting from this drug, then to perform a repeat or a new trial specific for that subset and prove it statistically with the numbers that would be required to rule out random variation. So that's a great question, but typically that's how it would proceed going forward.
Okay. And the next question would be looking at the opposite side of the coin there. When a trial drug performs so well, you discussed it leapfrogging through or fast tracking for approval. Who decides which drugs and when those drugs can go through that fast track process?
So that's a great question, and typically that is a combination of the sponsor, and usually that would be, you know, the commercial company that that owns the drug and has been running the trials, and the FDA, or in Europe the EMA, and a sit-down discussion to determine, you know, what these sometimes could be considered arbitrary numbers are. However, if it is an extreme benefit and it's clearly beneficial, then it's usually at a sit-down between the FDA and these the sponsor, and often they'll have principal investigators that were involved in the drug speak on behalf of the agent in favor of, for example, this fast track approval.
Okay. Well, thank you so much for the answers to those questions. As you know, this webinar was brought to you by Debbie's Dream Foundation, Curing Stomach Cancer. Thank you to our speaker, Dr. Daniel Catenacci of University of Chicago, and to Genentech for providing the funding that helped make this webinar possible. As you see on your screen, we have upcoming events to get involved in and support. Details about these events are available on the Debbie's Dream website at www.debbiesdream.org. Thank you to all of our listeners today. Our next clinical trials webinar will be held on November 4th at 12 noon Eastern Time. We would love to hear your feedback, questions, and thoughts before then. Please send your comments to patient.resource@debbiesdream.org. Thank you again.